I hope you can see my screen. Hello, everybody. Can you see my screen?
Now, yes.
Oh, okay, perfect. Okay, so first of all, thanks a lot, Claudia, for the very nice introduction. And I would like to thank Hira and all the organizer of the conference, because it's a pleasure for me to be in the conference and to have the opportunity to provide this talk. And I hope next time we will be back to the live conference. So today I will discuss the study we are performing, in which we are trying to characterize the genomic profile of cancer stem cells and to link the profile to the ability of cancer stem cells to modulate the susceptibility to immune responses. So I don't have anything to disclose. Just a brief introduction. It's well known that tumors are characterized by high heterogeneity in terms of genetic, molecular, and phenotypic characteristics of the cellular component. And in particular, the tumor comprises several cell clones coexist in the tumor lesion, and the generation of different cell clones is due to the genetic and epigenetic instability and diversity of the cells within the tumor that represent the driving mechanism towards cell transformation, tumorigenesis, and progression of the disease. And it's shown here in the cartoon that there are different components: the genetic diversity, the epigenetic and molecular pathway, together with the interaction of the tumor microenvironment and the component of cells with the with the stemness features that altogether this interaction lead to the clinical outcome of patients. And it's shown here that we have a genetic and epigenetic instability that from normal cells lead to the development of founder tumor cells. And from there, we have different clones that are developed upon the acquisition of different genetic and epigenetic mutation. So just briefly.
Cristina, I'm sorry, could you please make it the full screen, because we see only the slide, and yeah, sorry.
Can you see now the full screen?
Yeah, full screen, please.
Yes, now is the full screen. Can you see?
No, this is not. We see
it's taking a little bit of
on the left side, and therefore could you please open
because from my computer is full screen, so I don't
probably could be a problem of connection is slow.
Yeah,
try to go back to the PowerPoint and then again launch the presentation. Echo. Yes, it was.
What about now?
Yes, I believe it's just a little bit slow for the connection. If we wait 1 minute, probably we will have the full screen.
Okay, so let me know if it's working. Okay, so the cancer stem theory belongs to the 1997, when the group of John Dick was able to describe that indeed within a tumor exists a hierarchical organization, which cells endowed with the stemness property are the main player, the main responsible of tumorigenicity and tumor formation. And since then, this theory replaced the stochastic model in which all the tumors were supposed to be endowed with equal ability to generate tumors. So now it has been demonstrated that there are different clonal cells with different properties, and only the cells with, I mean, the tumor, the ability to generate and lead to the progression of tumor is mainly dependent on the presence of cells with stemness property. This is just to show the evolution of the theory of cancer stem cell, in which since 1997, subsequently different study demonstrated that indeed cells with the staminase property exist also in solid tumor, starting with the first publication 2003 for breast cancer, and then cancer stem cells has been isolated from.
Cristina, sorry, I have to stop because several people cannot see the full screen. Maybe you should just sharing the screen of the PowerPoint. Okay, now just, it was good just.
Because, okay, now I'm in just a regular PowerPoint.
Okay, and try to put in presentation again. If it will not work, you will do in a PowerPoint, just this.
So on my screen now I see on presentation.
Yes, but we have, what we see is cut. Okay.
How do you see now?
We see with the presenter from the speaker. So it's, we see all the pictures. So maybe it's better the screen of PowerPoint. Did you share the screen? You have to share the screen.
Yeah, yes, the share is, I mean, I'm going, okay, let me exit and try again. I'm sorry for that.
It was good. Okay, if this this is the best among the different, even if we see the slide on the left, maybe at least I can see most of in the screen most of the presentation. Ira, do you think it's okay like this? If she can not.
Yeah, Cristina tried to open and to provide the full screen, but yeah, perhaps click here, Cristina, and yeah, and perhaps we will see the full screen, but since it doesn't work, perhaps there is a problem with
Cristina, from time to time you press something that it shows correctly.
Yeah.
Sorry, say it again, please.
From time to time you press something that shows the screen correctly, full screen.
Yes.
So it can work, just...
When you go back to the PowerPoint, just one time it goes in the right screen and then go out through. So just try, stop.
No, the thing is, when you see the full screen is when I stop to sharing the screen. So it's something strange.
Okay, we have to continue because we are just
of the time
now.
Yeah.
So do you prefer I leave
like this?
Like this? Okay, I will leave like this. Okay, let me see if I can. Okay, it's better?
Yes, at least we can see all the
okay, so
all the slides.
Okay, as I was saying, so the cancer stem cell, when subsequently isolated by most of solid tumor with different histological type, and in particular, it has been shown, well demonstrated, that cancer stem cells are responsible or resistant to therapies, in particular chemotherapy and radiotherapy, and it appears they represent the substrate minimal residual disease upon the therapeutic treatment, in which, despite the initial responses by cancer patients, cancer stem cell resist to therapy, so survive inside the tumors, and then they can go back to give rise to start again to enter in the self-renewal phase and give rise to new clonal subpopulation of the tumors, and in which part undergo differentiation, so represent the bulk differentiated tumor cells with rare stem cells still present in the lesion. So based on that, so cancer stem cells have been isolated from based on the property of cell renewal, the multipotency, so the ability to give rise to the variety of differentiated cells found in the original malignancies, and also the tumorigenic ability to generate a phenocopy of original malignancy in immunocompromised mice. This is just a picture of our previous work in which we were able to isolate cancer stem cells from colorectal cancer patients. So the first row, you see how the cells, the cancer stem cell grow in the form of sphere, and the second part is the correspondent differentiated bulk tumors isolated from the same tissues. And here is showing the last column how cancer stem cells undergoing differentiation, for example, in the presence of fetal bovine serum, they start to differentiate, and the phenotype is similar to the one of the bulk tumor. We also characterize the expression of a marker that has been described to be associated with cancer stem cells. Up to now, we don't have a specific cancer stem cell marker available, allowing us to directly isolate ex vivo cancer stem cells. And so we use the ALDE floor to isolate that is overexpressed, shown to be overexpressed in cancer stem cell, to isolate from either our tumor sphere forming cells or our differentiated cells expressing ALDE floor. And indeed we characterize by cell sorting that cells overexpressing ALDE floor, like in the black and gray bar, also express higher level, superior level of markers such as CD44, 24, 133, EPCAM, and CA that have been shown to be overexpressing cancer stem cells from isolating from colorectal cancer. And we also demonstrated that upon the ex vivo isolation of cancer stem cells and the infusion in the transplantation in immune deficient mice, cancer stem cell, that is the top graph, have higher superior efficiency in forming tumors, even using 10 to the second cells as compared to the bulk tumor. And also the cancer stem cells is representing in the picture on the bottom part, left bottom part of the slide. The phenotype of cancer stem cells is more similar to the original tumor derived from the patients. So based on that, also is also important to note that there is a strict crosstalk between cancer stem cell and the tumor microenvironment. In particular, the tumor microenvironment represents the niche of the cancer stem cells. So normal cells reside in niche, and normal niche is comprised stromal cells, fibroblasts, immune cells, endothelium, perivascular cells, in addition to different soluble growth factor, cytokine, and also the extracellular matrix. Resembling this situation in the tumor microenvironment represents the niche of the cancer stem cells. And this has been shown there is, the tumor microenvironment is a complex entity in which there are different subpopulation of cells, and also a strict crosstalk and regulation among the different cells. And it has been shown that also the tumor microenvironment can play a survival and proliferation for cancer stem cells, and at the same time also cancer stem cell can play an important role under the development and differentiation of immune cells and regulating their function. So our question is, can indeed the available tools target cancer stem cell in an efficient way? This is a complex topics. I don't want to go too much into the details regarding the different approach. I have summarized here different approach commonly used for therapeutic treatment of cancer that have been designed with the aim to target molecules or aberrant signaling pathway in cancer stem cells. However, despite the PROSIMIS results, none of them showed to be efficient in completely eradicating the tumor. That means to target and kill all the cancer stem cells. And this is due to different mechanisms of resistance associated with the cancer stem cells. I'm just showing here, for example, for aldeflor that I mentioned before, the role of this molecule in inducing radio or chemo resistance for cancer stem cells. And here is a cartoon summarized with different mechanisms utilized by cancer stem cells to resist to the therapeutic targeting that include anti-apoptotic pathway, remaining in the quiescent form in order to avoid the targeting by different, for example, chemotherapy, the upregulation of pro-survival signaling, the induction of signaling that allow them to survive in hypoxic condition in the niche, the upregulation of DNA damage response, and so one of the question is how can we efficiently target cancer stem cells? So from this point of view, immunotherapy represents a promising approach. It has been shown that the immune system can control the development and the progression of the tumors, and shown here in this old publication in the context of melanoma, it has been shown that the presence of immune cells in the tumor lesion has a prognostic role and correlate with the survival of patients. Since then, different studies has been performed confirming this evidence, also more recently in a very elegant details a study performed by the group of Jérôme Gallon in France from colorectal cancer. However, since 2010, immunotherapy represents the fourth pillar of the treatment of cancer, thanks to the development of immune checkpoint blockade, which are methods to unleash the immune system to attack cancer cells by using agents that inhibit immunomodulated molecule expressed on either on the immune cells or the tumor cells. And this led to the recognition of a Nobel Prize to Tazuko Onyo on Jamie Allenso in 2018. And more recently, also a cell-based immunotherapy approach has been approved for the first time for the treatment of hematological disease that is based on the engineering of ex vivo engineering of immune cells with the receptor that can recognize and attack tumor cells. So based on these evidences, immunotherapy represents a promising system to target cancer stem cells. And that is being discussed by different groups and authors. However, it has also been shown that cancer stem cells can resist to immunotherapy. The initial study was published by the group of Lee in 2018, showing that upon a cancer vaccination in a mouse model, the cancer vaccination lead to a selection of cells that are resistant and are endowed with the stemness property. And also my team in 2010-2014 published data showing that indeed the cancer stem cells showed an immunoresistant profile and are resistant to attack by T cells. So we have performed a whole transcriptome analysis comparing cancer stem cells and differentiated tumor cells that here are denominated FBS. And it appears very clear there is a differential transcriptome profiling the two types of cells, and among the 469 genes that we found significantly differentially expressed, we could identify some molecules, some pathways that are associated with the immunological profile, such as some down modulation in the interferon signaling, TNF receptor, some cytokine, tumor growth factor, they also in receptor interleukin 1 were down modulated, such as the NKG2D ligands, such as MICA. But we found also gene associated with the antigen processing and the HLA expression, such as, for example, here I mentioned the proteasome, but we found also molecules associated with HLA that were down-regulated in cancer stem cells. And we confirm at the proteomic level, and I will show you just within a few minutes some results. On the other side, we found upregulation IL-4 receptor, and we indeed demonstrated in a previous publication that cancer stem cell for colorectal cancer upregulate the IL-4 receptor and also IL-4, and with this signaling, they are resistant to the attack by immune T cells. In addition to that, we found the differential expression of histone deacetylase, aldefluor, Hitchcock protein, and also the kynurenine hydrolase. And I will come back later for a few of these molecules. Here, I want just to mention some of the signaling we could identify. For example, we found a downmodulation of a mitochondrial dysfunction in cancer stem cells, and the other side, an upregulation of cholesterol biosynthesis. And here is one of the networks we could identify in which we that include some of the cytokines or growth factor I just mentioned, for example, IL-8, TGF-beta, interferon alpha, IL-12, that were differentially and downmodulated in cancer stem cells compared to the differentiated tumor cells. We also moved to the sorry. Can you still hear me?
Yes.
Oh, okay, sorry. And so we moved to the also to try to characterize the Mirna profile of cancer stem cells. And here also very interesting, we found a differential pathway, differential gene signature between Mirna in cancer stem cells that are indicated here as red, and the control cells that are the differentiated tumor cells. We then moved to we then move to the, we want also to validate these results by quantitative RT-PCR, and we focus on pairs. So the pairs of cancer stem cells compare to the differentiated tumor cells represented in blue, and by PCR analysis, we can also identify clearly different spatial distribution of the cells, and also is also clear here that, for example, there is a link anyway due to the patients, to the subject, between the two pairs of cancer stem cell and differentiated tumor cells. Anyway, by quantitative PCR, we found 67 Mirna that were differentially expressed between differentiated and tumor cells and cancer cells. In the table are summarized the 5 that were the top significant differentially expressed. We also did some analysis for the to identify the pathway by the ingenuity pathway analysis. I want just to mention here some Mirna such as that play also an important role in regulating immunological function. And so we found, for example, Mirna 21 that upregulate regulate the TGF-beta. Also, this Mirna is also involved in the regulation of some genes that are associated with the cancer stem cells, such as OCT4, NANOG, and SOX2. Is also involved in the regulation of STAT3, which is an important regulatory molecule for different immunological function, including the expression IL-12, P10, and also regulate also the epithelial to mesenchymal transduction pathway. We found also other Mirna listed here in which regulate the expression of chemokine receptor, miR33 can regulate autophagy, lysosomal mediated cholesterol biogenesis. And if you remember, I just showed the slide in which the cholesterol biogenesis was down more modulated in cancer. It was upregulated, sorry, in cancer stem cells. Also, this miR can regulate the expression of PD-L1 and also the activity of IDO, that is an enzyme involved in the degradation of tryptophan, and has been shown to play an immunoregulatory function in tumor cells. And then we have other miR regulating the expression of B7H3, TNF of interferon signal. So we have a large array of regulatory miR involved in the immunological the expression of immune-related molecules, and we found a link between these and also some gene expression that I showed before. This is just to show one of the network we could identify. In this case, I show also, I indicated also a network associated with the p53 molecule. That's also one of the relevant molecules in tumorigenesis and associated with the aberrantly expressed by cancer stem cells. We also use two different databases, TargetScan and MIR-DB, to compare the results obtained by our miR profile in order to validate target genes that are shared by the two databases. And here I'm showing here, for example, how just picking up 5 miR, how many target genes can be involved. And at the end, we can also come out with 8 target genes that are shared by these 5 miR. Anyway, I mean, from the functional point of view, we show and published previously that indeed some of the gene involved in the antigen processing and presentation of peptides of antigen in association with MSC class 1 or class 2 molecules were downregulated in cancer stem cells. And we also analyzed the expression of some ligands, NKG2D, that are ligand of the NKG2, the receptor that is associated, a co-stimulatory receptor for T cell and activatory receptor for NK cells. And indeed, we found a general down modulation of these molecules that is shown in these representative data by comparing cancer stem cell and differentiated tumor cells. And here is the detail in which we analyze a large array of molecules involved in antigen processing, the degradation of antigens and association with MSC molecules in order to present on the surface. And we found a general down modulation in cancer stem cell derived by glioblastoma, but similar results were obtained also for colorectal cancer. And for regarding the NKG2 ligands, we found, as shown here, that the level of expression BK, or BP were quite down modulated in the cancer stem cells. And on the other side, we found the presence of soluble molecules in the supernet on the cell lines, and it has been shown that the soluble molecule play a negative immunoregulatory role for immune responses. We also demonstrated that the co-culture of cancer stem cell with the T lymphocytes in this, this experiment was performed by co-culturing cancer stem cell or the differentiated tumor cells with the allogenic lymphocyte derived from a healthy donor. And we can see, compared to the specific mitogen stimulation and down modulation of a proliferation measured by CFCA and flow cytometry as compared to the differentiated tumor cells. And this was performed by using cancer stem cell and FBS of two different, isolating from two different patients. So in general, we found an immunosuppressing profile of cancer stem cell compared to the differentiated tumor cell, and shown by the ability to express MHC molecule, the NKG2D ligand, the modulation, the ability of interferon of demethylating antigen to modulate these molecules, and also the efficiency in inducing T cell responses upon co-culture with the cells. And we, for example, in the colorectal cancer, we demonstrate that indeed colorectal cancer cannot be efficiently recognized by T cells. On the other side, NK can efficiently recognize the cancer stem cells due to the efficient expression of activatory NK receptor. And here, for example, we recently started a collaboration with HERA to aim to characterize, for example, the immunological profile of cell lines. Here are is one triple negative breast cancer for which she was able to derive from the parental cell lines with the more higher invasive or radioresistant property. And we can see here that we can observe a general low expression of HLA molecules, or also class 2 molecules, ICAM1, and also two molecules that has been shown to be can play a role as antigen recognized by T cells, in addition to the immunoregulatory property of the 7H3. And we can see that, for example, in invasive and radioresistant cells show a superior downmodulation of the HLA molecules. And this was also shown for the different panel of molecule involving the antigen processing and machinery by performing intracytoplasmic staining of these molecules. And we try, for example, for this is another breast cancer cell line. This is a luminal A subtype in which we culture the cells under the condition of cancer stem cells. And for example, for the invasive cell lines, we could identify cells with the phenotype of cancer stem cell forming the sphere. We still need to characterize the expression of a specific marker. And we saw in some cases, for example, an additional a further downmodulation of HLA molecule as compared to the invasive culture in standard conditions. So we, our plan is to further characterize these cells in order to identify if they can they are endowed with some cancer stem cell molecules or cancer stem cell associated markers, and they can behave in some ways as cancer stem cells. And here, since I mentioned that we found the Mirna regulating an enzyme involving the degradation of kynurenine, that is the pathway mediated by IDO. As I mentioned, IDO play an immunoregulatory function, since can degrade the tryptophan to kynurenine. And indeed, we demonstrated that upon co-culture of cancer, we demonstrated, first of all, that the cancer stem cells, upon treatment with interferon, that is a molecule that can upregulate IDO, it has been shown also in the lesion of different type of tumor, cancer stem cells express superior level of kynurenine as compared to the differentiated tumor cell pair. And this is true for glioblastoma, but also for the colorectal cancer, shown in the panel B. And here is also a picture in which we perform the staining for the expression of IDO by confocal microscopy, and we could decrease the expression of IDO upon the treatment with the specific inhibitor 1-methyltryptophan. And here, for example, we can observe that upon co-culture of cancer stem cells, either with the autologous lymphocytes, with either the differentiated tumor cells of the cancer stem cells in the line in the bottom part of the slide, indeed there is a superior inhibition of proliferation, still measured by CSCs, in which proliferative cells divide the fluorochrome, so are less stained with the fluorochrome, and compared to the differentiated tumor cells. And this is upregulated by interferon in line with the what I showed before, that the expression of IDO is superior in cancer stem cell treated with interferon compared to the differentiated tumor cells, but also we can rescue completely the ability to proliferate if we inhibit IDO by treating the cells with the 1-methyltryptophan specific inhibitor. And in addition, so we could demonstrate that we have a general immunosuppressive phenotype in cancer stem cells compared to the differentiated tumor cells, and we could identify some Mirna that can regulate. So we are currently performing some functional assay in order to prove the role of Mirna for the immunosuppressive property that I just showed with the functional in vitro experiment. But in addition to that, I just want to briefly show you something we are trying to demonstrate. So it has been shown that the epithelial to mesenchymal transition is a mechanism that occurs in tissue that undergo chronic inflammation, and also play a role in the development of cancer stem cells, since it is commonly found in tumors, in tumor lesion, and is the mechanism that promotes the invasion of the cells, the extravasion and the systemic dissemination of the cells, leading to the colonization of secondary organ and the formation of secondary tumor. And here is the summary of the molecule, the pathway involved in the regulation of the epithelial to mesenchymal transition. And it has also been shown that there are epigenetic mechanisms that control epithelial to mesenchymal transition. So what we are trying to investigate, and I'm just showing you some preliminary results, is that we are focusing on the patient with inflammatory bowel disease, since this patient, depending on the duration of and the severity of the disease, are more susceptible to develop a colorectal cancer. And here is the cartoon showing what happens in the tissue upon chronic inflammation. There is a disruption of the structure of the epithelium in association with the change of the population of immune cells that are present in the tissue, leading to a chronic inflammation. And here is what happened, the evolution of IBD from normal colon to colitis, indefinite dysplasia, the development dysplasia that is associated to the some specific, also the occurrence of specific mutation of a molecular aberrant expression of some specific molecules such as STAT3, TNF, the down modulation of the DNA repair, followed by mutation in KRAS and APC, leading to high-grade dysplasia and then the development of carcinoma. So some of the genes are involved also in the development of sporadic colon, but the sequence of the mutation is different in patients with IBD. And so what our point is to try to demonstrate that we have a sort of dormancy of a tumor originating of cells originating the tumors, and this can be identified in earlier stage in patients with chronic inflammation such as inflammatory bowel disease. So we have performed, for example, a comparison of pediatric versus adult patient with IBD. Assuming that adult patient with long-term history of IBD can be more susceptible to the development of colorectal cancer. And our point is to identify genes involved in the epithelial to mesenchymal transition, and possibly involved also with in early stage development of stemness signature. And we found Mirna differentially expressed in pediatric versus adult patients. Just here, I want to mention some of the top differentially expressed Mirna that indeed are associated with the epithelial to mesenchymal transition. All these Mirna regulate this mechanism, in addition to some immunological mediated responses, such as the presence of IL-23, the presence of Th17 cells. But I mean, surprisingly, I mean, we are very surprised to find that just in the initial experiment, we could identify miRNA-regulated EMT, and also involved in also in autophagy. And here, for example, is a bigger, more comprehensive picture of the miRNA we found differentially expressed in pediatric versus adult patients, some of this miRNA has been also shown demonstrated to target gene that are shown here in the cartoon, ATG16L1, NOT2, IRGM, involved with susceptibility to the development of IBD, as well as immunological mechanism. And in addition to that, we are trying to characterize the differential methylation profile of the tissue in adult versus pediatric IBD. And indeed, we found, these are very preliminary results, a differential methylation profile in these patients, and which regulated some of the island regulated encode for gene that has been associated with the development of colorectal cancer, with the EMT phenomenon, and also with some genes that are associated with the stem. So we are still working on this. These were very preliminary results, but this seems that are promising, and with this we want to show that we can identify a link between the chronic inflammation and the early generation of stemness signaling in patients that can be susceptible to the development of colorectal cancer. And this here is just a cartoon summarizing what has been defined to be the immunological and tumor dormancy, in which in the initial formation of tumor, the immune response can indeed effectively target cancer cells, including and this can be targeted by T cell or NK cells, but then there is a subpopulation of cancer stem cells which are immunological resistance, and they evade from the recognition of immune cells, and they can also promote the differentiation of the cells that are regulatory or inhibitory of immune responses, such as T regulatory cells, monocyte, inhibitory monocyte, myeloid-derived suppressor cells, or immunosuppressive dendritic cells, and these evade from the immune response, and so they can proliferate at a later stage, lead to tumor recurrence or progression. So there is a lot of we have a lot of mechanisms involved in the resistance of cancer stem cells to different therapies, in this including immunotherapy. And here I just cartoon summarize the complexity of the tumor due to the high grade of heterogeneity and the presence of cancer stem cells that can evade the immune responses that can lead to the progression of the tumor. And so we need to take into account all the factors, in particular also the interaction of tumors with the tumor microenvironment, in addition also to an interpatient diversity. So in conclusion, cancer stem cells or cancer-initiating spread display low immunogenic profile with suboptimal expression of HLA or antigen processing machinery molecule. In some cases also they can downregulate the NKG2D ligands, leading to impairment of T cells, but in some cases, depending on the immunological profile, also on NK cell mediated responses. We demonstrated that can be resistant to the recognition by T cells. And we have identified differential gene expression profile and miRNA profile that can regulate the immunological profile of cancer stem cell. We are in the phase of validating from the functional point of view the role of miRNA or some of identified miRNA in this mechanism. All these properties drive immunological dormancy of cancer stem cells. So we need to further investigate and better investigate this topic in order to identify the specific mechanisms that lead to the immunoresistance and try to overcome this resistance by implementing, for example, the treatment with immunotherapy or development also the combinatorial mechanism that can lead to the resistance. Here is just a cartoon, summarize all the methods, I mean, the immunotherapy approaches and how we could implement by, for example, combining with the epigenetic modification, with the chemotherapy, radiotherapy, target therapy, targeting specific pathway or even specific molecules involved in the resistance of cancer stem cells to therapy. And finally, I want to thank all the people involved in the study. First of all, my team here at Sidra Medicine. It is a laboratory of immunobiological therapy, Eva, Shilpa, Moza, Saad, and Aspa. Unfortunately, some of them just recently left Sidra to reach other institutes. Then the Omicron facility for all the genomic analysis, the system biology department for the bioinformatic part. I would like to thank the group collaborating to the study, the team of Dr. Said Dermime at Ahmad Medical Corporation, Professor Soldano Forrone at Harvard University, the group of the University of Palermo, Matilde Todar and Giorgio Tassi, and also IRA for the previous collaboration and also interesting discussion. And I also want to thank the previous group where I used to work under the lead of Professor Parmiani for the study that I show you a few of the results were performed at the time, in addition to the group at Ena Wang and Franco Marincola. And I want also to form all the collaborators here at Sidra, allowing the collection of samples. So the gastroenterology and the pathology, both at Sidra and the Ahmad Medical Corporation for the IBD study. And I would like to thank you for the listening to my presentation. And I will be happy to take any further question. Thank you.
Thank you, Cristina. A wonderful talk, very interesting. I think is right also interesting new insight and question. And please, you can directly ask by voice. This could be nice, and you can also write on the chat the question. So first of all, I can say that the general approach now is really to have an approach of combination of different therapies, because we don't have a specific markers. We have a plethora of markers, a different pathways that are involved, immune system and the cancer cells that are involved. So only the combination seems also by your data the most fruitful approach.
Yes, I mean, I fully agree. I mean, since the development, for example, development of the initial approach of immunotherapy, now we made a lot of progress, and there are, I think, hundreds of clinical studies in which we are trying to assess the combination of the different approaches, even the combination of immunotherapy with radiotherapy or chemotherapy, because it has been shown that these two standard care therapy can upregulate the immunological profile of tumor cells. I mean, we didn't join yet the final conclusion in which combination is the most effective, but I think what it has been in some way neglected up to now was to consider the component of stemness. So immunotherapy was not specifically taking in consideration the presence of stemness, of cells with stemness property. And so we don't know exactly, I mean, up to now, the development of immunotherapy was designed based on the bulk tumor, not considering the component of tumors that are resistant. So I think we need a further study to address clearly and in well manner this topic in order to implement the usage of immunotherapy.
Okay, thank you. We have two questions, one from Miria Castillo Martin. She asked about which biomarkers do you use to identify colorectal cancer cell in the whole population. You show that you have a plethora in a way.
Regarding the marker, I mean, I show that we are using aldefluor, but in association with different molecules, like for example, LGR5, that has been shown to be specifically expressed in stem cells from colorectal cancer, as well as a CA, EPCAM, CD44. So different, I mean, we are using a combination of markers to make sure we are not leaving out some or subpopulation of stem cells.
Okay, thank you. From Bruno Sainz, great talk, Cristina. Regarding Mirnas, were there were any Mirnas of interest that were downregulated, for example, in pancreatic cancer stem cell? He shown that the regulation of Mirna 1792 cluster.
Yeah, yes, we found, I mean, we found different Mirna, some up regulating and some down modulated. So right now, I mean, we have selected two of them. So our approach is to consider the one down modulating, they also up regulated in order to assess which one can play a role in the immunosuppressive profile of cancer stem cells. Okay,
thank you. Anna asked if in transcriptomic analysis of cancer stem cell versus FPS treated and differentiated treated tumor cell, did you observe any difference in the profile for the proteolytic enzyme? If you check something about proteolytic enzymes.
We didn't focus on this specific category of protein. I mean, I can for sure try to check. I mean, it's an interesting suggestion because we were more focused on protein. I mean, on the expression of molecules that play a role in antigen presentation, so directly or cytokines, growth factor. But I think it's a good suggestion, and we can go back and check it.
Okay, thank you. We have to go ahead because of the time. Everybody can anyway keep in contact with the presenters. So we can come back then in another time to